too.
So the first thing I want to say is that today's Wednesday. I just told Ms. Vininger that I'm going to text her when class is done and let her know when she should be expecting you guys at Rye High. And I'm going to do that for the rest of the semester on Wednesdays. The other thing is that the way that the schedule works, unless there's a major power outage on Monday or something like that, we are going to be scheduled for having a quiz on Tuesday, and that quiz is on this chapter. We're going to finish a third of this chapter today. We just finished a third yesterday, and then we'll finish the last third on Monday. Maybe we'll do a little bit more today, I'm not sure. In terms of the chapter itself, we talked about, there's three major topics. We talked about the basics of the nervous system, the structure, the overall structure of the nervous system, and then we talked about different types of cells. In particular, we talked about neuroglia or glial cells and neurons. Talked about where the neuroglia are found, what their overall functions are, and how they contribute to the overall health and well-being of the neurons. For the rest of the lecture, we're not going to, for today and Monday, we're not going to talk about neuroglia at all. We're going to mention them in passing once in a while, but they're not really going to play a role here because we're talking about two things now. First is today we're going to talk about action potentials. What's the structure of an action, or the overall layout of an action potential? Where does it occur? What happens to the membrane? How does the membrane react? And what happens immediately after the action potential passes through? That's what we're talking about today. And then on Monday, we'll talk about the structure of synapses, because if you have an action potential and it goes all the way down to the axon terminal and then nothing gets propagated further, then that signal is lost or nothing happens. So that has to propagate beyond the extent of the neuron, and there's a number of different things that can happen, one of which we've already talked about this semester, and that is skeletal muscle contractions. So that neuromuscular junction, if the signal gets sent to the muscle and the muscle doesn't respond, then that signal is lost. So that's if the muscle does respond, then the muscle contracts. On Monday, we're going to talk about how some of these neurons will be what's called so the abbreviations will be IPSP or EPSP, and the PSP is postsynaptic potential, and we've already talked about the presynaptic neuron, and now we're going to start talking about the postsynaptic neuron. And the I is either inhibitory postsynaptic potential or excitatory postsynaptic potential. So the strength of a given signal or the frequency of a given signal either could be dampened or magnified. And if it's magnified, then a lot more happens. If it's dampened, then a lot less happens. So that's where we're headed in this lecture. And so let's get into it. Here is where we ended yesterday. We talked about the distinctions between gray and white matter. We talked about nerve tracts versus and nuclei, ganglia, and nerves, whether they're being in the central nervous system or the peripheral nervous system. The vocabulary that I used yesterday was new, except for when I talked about it last week in my diagramming and stuff like that. This is, let me see here. Some of this is things that we've already talked about. At the top, I just put this slide right here. It's later on in this lecture series. At the top, we see a neuron with a multipolar neuron with dendrites extending more or less to the left, and then a single axon extending to the right. At the end of that axon, there's a synaptic. There's a synapse. Sorry. Those synapses are. We're going to talk about what happens here in this part right here. This will be for Monday. So for today, all we're doing is talking about what's happening from here at the axon hillock all the way down here. So that's what we're doing. The other thing to recognize is that the way this axon is drawn, it's much, much more complicated than that, because the axon has branches off of that, and then the branches themselves have branches. So a branch off of the axon is called a telodendria, and a branch off the telodendria is then well, there's a number of different things, but finally the last thing is called a terminal axon. So this right here has just a terminal axon drawn, a single terminal axon. Another thing to look at, regardless of what we're talking about and how detailed we get into it, is that all we're talking about here is the movement of ions across this phospholipid bilayer, and that movement of ions, when it is getting set up, it's active, but when it's happening, it's passive. Okay. And it's really important to distinguish that, because if you don't understand that, then you're going to be, you're going to get confused and you're going to get the answers wrong. So the other thing to recognize is that this is where my pointer is just a phospholipid bilayer, and we'll talk about what that is, we'll remember what that is in a minute. And then finally, there's a series of action potentials. So my pointer right here will, if in a simplified version of this action potential, the action potential will happen here, and then it'll happen here, and then it'll happen here, and then it'll happen here, and so on and so on and so on. If this axon were myelinated, then it would happen only at the nodes, and it would jump, it would be saltatory, it would jump from one to the next to the next. In this case, because there's no myelin present, this is a continuous action potential. This continuous action potential would be slow, relatively speaking, and it would, and once it starts, it's going to go to its end, and that end is at the terminal axon. So there might be, I don't know how many, there might be hundreds or thousands of action potentials that are observed at each individual spot on this axolemma. The cell membrane of the axon is called the axolemma. The axoplasm is the cytosol within the axon, and the action potential there might be many of these. So for each spot on this axolemma, if you look at the graph below, this is what happens. In order for the axon to get ready to send a signal, an action potential, it has to be at a resting membrane potential of negative 70 millivolts. If it's at anything less than, or more than that rather, it's not going to send anything. So in other words, if it's where my pointer is now, it won't send a signal. It's simply not negative enough. The analogy that I've drawn right here is when this pointer is pointing right there, this is a set mousetrap, and the mousetrap is down in my basement. It's got lots of peanut butter on it, and I'm trying to kill a mouse. So that's what it's set at right there. Without everything, all that prior work, that capture of that mouse, that killing of the mouse doesn't happen. Without all the prior work here, the action potential doesn't happen. When the signal gets sent, when that signal gets sent first to the dendrite, then to the soma or the cell body, and finally accumulates in the axon hillock. When it accumulates in the axon hillock and then gets sent down the axon, the first thing that it has to do is it has to reach a certain level. That level is called the threshold. If it exceeds that threshold level, that is to say the signal, if the signal exceeds that threshold level, then that signal will be propagated. If it does not exceed that level, then nothing happens. If you wanted to test this out, by the way, so I just see several of you with AirPods in your ears. Can you just do me a favor and take those out? It doesn't look good. A signal. You know, let's just talk about a threshold signal. If you're sitting next to somebody right there, and you wanted to touch their ear just enough so that they barely feel it, what are they going to do? Now, yeah, I knew someone would do that. Thanks, Amaya. I knew someone would do that. I was testing you, and you passed the test. Okay, you got you're listening to me. I know. But the point here is that if someone barely touches your ear, you don't feel it, right? Or you don't react to it. You might feel it, but you don't react to it. Then if someone grabs the ear and pulls on it, right? Then you say, wait a minute, stop. The signal, wait a minute here, wait. Stop beating up Arlen, okay? Those differences in signals here, that very, very gentle touch is below the threshold, no response. The touch that's more severe or more significant exceeds the threshold,
and then that causes what's called depolarization. That depolarization occurs at the axial lemma, and then immediately after that, repolarization, hyperpolarization, and then reestablishment of the resting membrane potential. So there is this stimulus right here. That stimulus can be either graded or it can be or not graded, right? So graded means gently increasing, or it can be just increased magnitude. So in other words, if I were to go over to Alan and grab his ear and pull on it, right, that would not be a graded potential, right? That would be just a major thing. Alan, can I try that on you? No? All right. I just wanted to see if you were listening. So these areas of the graph are characteristic. They need to be described by you and understood by you. And let's talk about what happens at each of these areas in the axon, in the axial lemma, and how it proceeds. So these electrical signals are called action potentials. The membrane potential, or the resting membrane potential, has to be established prior to the action potential getting propagated. If it's not established, once again, nothing happens. The ionic concentration of the resting membrane potential has to be established. And what are we talking about here? We're talking about movement across the plasma membrane. We're going to use a different term called the axial lemma, but we're moving across the plasma membrane. When you're forcing something across the plasma membrane into a higher concentration, that's called active transport. This process that we've already talked about, active transport, we talked about passive transport and diffusion in the cell slides, or cell chapter rather. And one example, the example that I used in the first unit, was the sodium-potassium pump. The sodium-potassium pump is is illustrated by this graph right here, or this slide. That red line right there is that I added to this slide is the phospholipid bilayer, or in this case the axial lemma. There's the liquid outside of the cell, or plasma, extracellular fluid, intracellular fluid, axoplasm, or cytosol, all of which are the same thing. The concentration of the sodium is higher outside the cell than inside the cell, much, much higher. How is that established? Through active transport. Similarly, the concentration of potassium is much, much higher inside the cell than outside the cell. And how is that established? Again, through active transport. Now, if the door is closed, if the doors to this phospholipid bilayer are closed, and nothing is permitted to pass through this, then this doesn't get upset. In other words, the movement of the ions doesn't happen. Once the proteins open up, once the doors open up, then the sodium will move into the cell and the potassium will move out of the cell. And that's the way it happens, actually. First the sodium moves in and then the potassium moves out. All the while it changes the resting membrane potential. The inside the cell, it's very, very negative. How negative? It's negative 70 millivolts. The concentration of negative ions on the right-hand side, including these proteins right here that are assembled within the cell. These negative ions are much higher concentration, and that, again, that causes that resting membrane potential. You can see some questions on the right-hand side of this slide, and they sort of illustrate what I just talked about. This is another slide that I used from chapter 3, the red dotted line. In the previous slide, what did that represent? This is what it represents. If you look over here on the left-hand side, there's a protein channel, or a transport protein. Those transport proteins might have receptors, and those receptors could be sticking off of these things. Those receptors could be carbohydrates, they could be other things. Right? Those receptors can be highly specific. They might be called glycoproteins, like I'm pointing out right here. Regardless of what we're talking about here, at the end of the day, these things are just integral proteins. These integral proteins will allow the passage, under the right conditions, they'll allow the passage of certain ions either into or out of the cell. If you, again, now look at this cell here, how would you tell, if I were to take all the labels off, how would you tell what was inside the cell and outside of the cell? Well, do you see these yellow lines right here? These yellow lines are filaments of the cytoskeleton. And these are filaments that are found that help stabilize the axolemma and the axoplasm and help strengthen the axon itself. Right, so these filaments here, general filaments of the cytoplasm, or the cytoskeleton rather is what it's labeled. These filaments are really, really important, and they are built within the soma of the neuron. They're sent down the soma of the neuron, down the axon, and that's called axonal transport. And that helps stabilize and strengthen the axon. So how would you tell that? If you look, you need to look from one side to the other for cytoskeletal elements. So we're going to get in depth into the sodium-potassium pump. As I said just a little while in the previous slide, there are the high concentrations of sodium outside the cell, high concentrations of potassium inside the cell, and that steep concentration gradient results when the proteins open, when the receptors open, or the channels open rather. Sodium moves in one direction, potassium moves in the opposite direction. When you synthesize something inside the cell that's too big to move out of the cell, in particular proteins, and then that helps negatively charge the cell, or charge it negatively. So those are the proteins that are assembled within the cell. Chloride ions are also present inside the cell, and there are a number of different channels open. There are some things called gated ion channels, and a gated ion channel is an ion channel with a receptor or a bouncer, if you will, at the door. If that bouncer or that gate allows something to go through, then it only allows certain things. There's also leak ion channels, and a leak channel, or a non-gated channel in this case, a non-gated channel will be just an open doorway. That open doorway will allow things to pass passively one direction or another. How is it going to pass? It's due to a concentration gradient. What's the process by which this passage occurs of the chloride ions in the non-gated channels? The process right there is called diffusion. Okay, so we've talked about active transport setting up the resting membrane potential. And now when you're talking about the movement, the passive movement of, in this case, chloride ions, you're just talking about diffusion. When the sodium-potassium pump activates, it pumps 3 sodium ions outside of the cell and 2 potassium into the cell. For each use, for each round of the consumption of an ATP molecule. And once again, this has to happen before the action potential happens. I mentioned different types of channels, gated ion channels. I'm also going to use the word ligand gated channel. And in this case, I mentioned also a leak ion channel. And leak ion channels, again, the doors are open. They don't allow a lot of ions out, but these things are always open. And what they do is they just allow for the difference between the inside the cell, the negativity inside the cell and outside the cell. It equilibrates. It sort of stabilizes at a particular level. There are lots of leak ion channels for potassium and chloride than sodium. And that's just a general pattern there. And this should be a leak ion channel. Oh, brother. Let's see here. Let's not reboot now. This should be a leak ion channel, not a lean ion channel. And again, these leak ion channels help maintain the resting membrane potential. So there are these leak channels, these gated channels, and these gated channels can be ligand, and then they could also be electrochemical. A ligand simply means it's a response to a chemical, and most often when we're talking about a ligand here, we're talking about a neurotransmitter. So a gated ion channel is a channel that just responds to a particular stimulus. It only responds to that stimulus. If another stimulus comes in and it's a different chemical, then it's not responding. So that ligand gated channel is going to be causing a response. In some cases, I mentioned earlier today, there's excitatory postsynaptic potentials and inhibitory postsynaptic potentials. A ligand or a chemical doesn't always have to open a gate. It can close a gate too, and if it closes a gate, then that could be an inhibitory response. Okay, so I want you to just sort of think about that as we go forward here. Once again, this term ligand is simply a molecule or a chemical that sticks to a particular molecule. Most of these are glycoproteins. I mentioned that in the previous, one of the previous slides. I've already talked about some examples of neurotransmitters, one of which is ACH or acetylcholine. We're going to talk about different types of receptors in the brain section here when we get to neurotransmitters. But acetylcholine is one example of a receptor. It has a particular affinity for a sodium channel, and so acetylcholine will cause the sodium channel to open. The sodium channel will then open and allow sodium into the cell. And why do I know that? Because I know that the concentration of sodium is very, very high outside of the cell. So as a result of sodium rushing into the cell, that changes the resting membrane potential, and then other things happen. This term right here, and this is another transmitter, serotonin. For this exam and for this quiz, I'm not going to ask you about different types of neurotransmitters. I will only ask you about the general ligand-gated, or one example of which is acetylcholine. So neurotransmitters there's a whole class of molecules, there's a bunch of them, and once again, we'll talk about those later on when we get to the brain. So we've talked about our leak channels, ligand-gated channels here, gated channels in general, and those are chemical, and then there's voltage-gated channels. A voltage-gated channel is one that's not responded to a chemical necessarily, it's responded to electrical stimulus. And that electrical stimulus is either when it responds there, or when that stimulus changes, it either opens or closes, again, in response, depending on what we're talking about here. If these are excitatory synapses here, then they're going to respond by opening. If they're inhibitory, then they're going to respond by closing. Most common voltage-gated channels that we're talking about here are sodium and potassium channels, and in the heart, which we're really not going to talk about very much, and I don't spend a lot of time talking about the heart and smooth muscle. Calcium is also very important. There's other channels as well. So there's touch receptors and temperature receptors. These can be receptors that are modified epithelial cells. We'll talk more about this again in the Unit 5. These modified epithelial cells will respond to a particular type of stimulation. If they're mechanical, respond to mechanical stimulation. If they're temperature receptors, they respond to the different changes or elevation or decrease in temperature. It says here that temperature receptors often are found in the skin, and there's also other internal temperature receptors as well. And those basically tell you whether in your body whether or not you have a fever. So there's different types of channels, and those channels respond to specific stimuli. If you put increased temperature at a touch receptor and there's no other temperature receptors around, you're not gonna detect it. If you now, that's a caveat here. There's lots of receptors everywhere, so that's sort of artificial here. Similarly, if I touch a temperature receptor and there's no touch receptors around, I'm not going to have a response either. If the signal doesn't match with the receptor, then there's no response. So we talked about this uneven distribution of anions and cations across this intracellular or extracellular fluid, and that's called the resting membrane potential. The resting membrane potential, I'm going to say, is -70 millivolts, but in practice, there's other things as well. It can be less than that. And once again, look at these numbers here. If I say it's less than that, I don't mean closer to 0, right? I mean further away from 0 because they're negative numbers. So don't get confused with that, and I'm definitely going to try to confuse you. Okay? So if this is, the resting membrane potential is more negative, then it's further away from 0, and so that would be negative 90 millivolts. But that area right here between 70 and 90 millivolts is about what we're talking about. For the purposes of this class, I'm going to say it's just 70 millivolts, and that's really just to keep it simplified. That resting membrane potential is established by the sodium-potassium pump, and it's in an unstimulated cell or unstimulated neuron. So we've talked about different types of channels here, and if the neuron is resting and there's nothing happening to it, then what's going to happen here is that there are things that are going to be called leakage channel or leak channels. Those leak ion channels will allow for the general movement of sodium into the cell and potassium out of the cell, and it basically just reduces the difference between the polarity of the cell a little bit. And what's going to happen then is that to maintain that negative 70 millivolts, the sodium-potassium pump, that active transport, helps maintain that. So again, this 3:2 ratio here, sodium to potassium, is diagnostic of the sodium-potassium pump. And now here we have a couple of different pictures of basically of the sodium-potassium pump. In this case, now, how would I know this cell or this diagram if I take all of so if I take this term cytoplasm, and really it should be axoplasm, because we're talking about the axonal and this ECF or extracellular fluid. If I took those two things out, how should you be able to recognize what was inside the cell and outside the cell? Well, first of all, I'd have to leave the labels here. So this pink and purple here would be sodium, potassium, respectively. There's a leak channels, these leak channels right there, right? That would be labeled there. You don't know which way they're going to go. One characteristic, though, internal, inside the cell itself builds negatively charged proteins that can't get out of the cell. So if you see this PR negative, that's a negatively charged protein. That green blob right there that my pointer is at means that by definition that's got to be inside the cell. We don't have floating negatively charged proteins floating around in our body or in our plasma. That would be grabbed by phagocytic cells and would be decomposed, would be broken down. So these proteins here, so how would you know that? Well, first of all, the proteins are higher concentration inside the cell than outside the cell. The second thing is that the potassium is higher inside the cell than outside the cell. And then finally, the sodium is concentration higher outside of the cell, inside the cell. So how would you tell, without me labeling it, what is what? Those are 3 points. And when you're talking about the movement of ions here, we're talking about the movement of leak ions here. This is the movement of the leak ions, or the leak channels there, is simply going to be dependent on the concentration gradient of the individual ions. It's not the entire number of ions. In other words, it's not the number of ions, negative ions, or positive ions outside the cell compared to inside the cell. It's the number of potassium ions outside the cell compared to potassium ions inside the cell, and the number of sodium ions inside the cell compared to sodium ions outside the cell. So you only count sodium to sodium and potassium to potassium. As a result, you know that you should be able to see or predict which direction the sodium and potassium will move. In this case, this is talking about moving, I think these are potassium ions moving out of the cell. And generally speaking here, what's going to happen if the leak ion channels are the only things functioning, is going to go from a negative 70 millivolt to a 0 resting membrane potential. Why is that? Because by diffusion here, you're going to have the equilibration, or the movement of sodium into the cell and potassium out of the cell until there's no difference. So without the resting membrane, without the sodium potassium pump, then that's what's going to happen. When you're talking about equilibrium here, the equilibrium is simply a match of the amount of, say, sodium moving into the cell versus sodium getting exported from the cell. The equilibrium is going to be based on the diffusion rate of these leak ion channels of either sodium or potassium, and compared to versus the sodium potassium pump. So that's how that equilibrium is set up. So this sodium potassium pump maintains the resting ion levels here, and that resting ion level then is really this. That is what it's happening here. So while the leak ion channels are opening, allowing the passage a little bit, the sodium potassium pump, and you can see it drawn right here on the right hand side, the sodium potassium pump is consuming ATP and it's doing it a lot faster. Because there's so many negatively charged ions inside the cell than outside of the cell, the inside the cell is more negatively charged, and that's that resting membrane potential. So if you look at this table right here, I'm not going to go through all of these things here, but these characteristics responsible for a resting membrane potential here. The first thing is that we have the concentration of potassium along with their leak ion channels, and then most importantly, you have to have these plasma membranes that's semi-permeable, or in this case, is impermeable to big things in the cell. So a plasma membrane, or the axolemma, or the axoplasm is, the axolemma is impermeable to big things, but it's still semi-permeable to other things. So in other words, it allows some things to move in and out freely, and other things it does not allow to pass through. These large negatively charged proteins are trapped inside the cell. When you're talking about leak ion channels here, you're talking about diffusion of either potassium or sodium. And that passive process happens continuously. And just sort of at the bottom here, at equilibrium, very little movement of sodium or other ions, so of potassium or sodium, takes place across the plasma membrane here. Very limited relative movement here. There's going to be, if you were to track an individual ion, you would see it going from one side of the plasma membrane to another. But the concentration gradient isn't going to change very much. That's what that means. And so because the concentration gradient doesn't change, the resting membrane potential doesn't change appreciably. So now let's talk about what happens at a particular spot in the axolemma. And the first thing that happens is depolarization. Depolarization occurs when that signal gets passed down the axon hillock, and the first thing that happens is that sodium ions are the first to be affected. Sodium channels open, and sodium diffuses down its concentration gradient, so that's diffusion, and the inside of the cell becomes more positive. So as a result of that, so that we go from negative 70 millivolts, we increase dramatically to approximately positive 30 millivolts, and then what happens after that? There's also calcium that will also affect this, and I'm not nearly as worried about calcium in this context here, except when we're talking about synapses here. So when we get into synapses later on in this chapter here, we'll bring up calcium later. once again, potassium is going to be found greater concentration inside the cell because of the sodium potassium pump. And that depolarization is going to change the ability of the ion here. When you talk about the concentration gradient here, we're talking about the receptors. The channels open in the potassium when the concentration gradient or the resting membrane potential gets too positive. The channels then open into potassium, and the potassium then rushes out. And as a result of that, the thing that as the potassium rushes out, the charge then becomes more negative, and as the charge inside the cell becomes more negative, it basically overshoots its mark. It overshoots its mark, and that, as a result of that, it's called hyperpolarization. So the two main functions or characteristics of why that happens here. If the channels are open, the potassium channels are open, there's potassium that diffuses out of the cell. I just said that. If there's too much of those potassium ions that move out, then the concentration gradient gets less than negative 70 millivolts. In addition, there are also leak chloride channels here, and those leak channels and some ligand gated channels there can also cause chloride to move into the cell, and if it's moving into the cell, again, that causes the cell to be negatively charged. So what happens as a result of that? Now, when we're talking about the whole length of this entire graph, that first graph that I showed you, this is talking about at a particular point in a neuron for maybe 1 to 2 milliseconds, or 1 to 2 thousandth of a second. This hyperpolarization, so that if you look on the x-axis right here, this time right here is only a couple of milliseconds here. So it's not very much at all. This is area right here where my pointer is, it would be defined as hyperpolarization. And again, the two things that are largely a function of that is too many potassium ions moving out and too many chloride ions moving in. And here is where we started with this discussion right here. This, again, now looking at this graph right here, we have this threshold limit reached here. As a result of that, the signal being above that certain threshold here. And what's that signal? That signal is ACH, or a neurotransmitter, that ligand that is binding to a particular receptor. You have the rushing in of sodium ions in depolarization, and the rushing out of potassium, and the rushing in of chloride ions during hyperpolarization. So those two things happen during repolarization and hyperpolarization. And then how does that respond? How is that reestablish that resting membrane potential? Well, the sodium potassium pump, we've already described that, pumping the sodium potassium in opposite directions. And then remember too, if you have chloride, if you're worried about chloride ions, and we will be, because we've been talking about them, the negatively charged proteins aren't going to move, the chloride ions are. There's leak chloride channels here that will allow, if the concentration of chloride is too great inside the cell, it will leak out of the cell so that the concentration of chloride inside the cell versus outside the cell is the same. So as a result of that, hyperpolarization is not a permanent structure or even a long-term structure. It's very, very temporary, and that resting membrane potential is reestablished. So now let's, before I forget, let me see here. So I see some people getting restless. This is sort of dense material, so it's about 20 after. So I think what you guys want to do, I want you to do is to take 10 minutes, come back at 9:30.